Right. So when the first round of the Human Genome Project was wrapping up, there was a lot of soul searching about the apparently small number of human genes in light of our apparent amazing complexity as humans. But luckily, at the same time, people realized that most human genes can be spliced into more than one transcript isoform. And I think that thinking of transcriptome size as the definition of complexity is a really facile explanation. So that doesn't exactly address the concern in this New York Times headline that I thought was sort of funny at the time. But it is true that alternative splicing adds a huge amount of complexity to gene expression. And one of the amazing tools that we have access to now for studying complexity and how the transcriptome defines cell types and cell states is single-cell RNA sequencing, which can, of course, reveal all sorts of things about cells. But until recently, it's been relatively hard to understand the impact of alternative splicing on the transcriptomes as studied by single-cell RNA-seq for a lot of reasons, mostly technical, to be honest. And so we've started over the last years trying to dig into this and see what we can do to pull some of the signal out of the noise. So before I get into the content of what I'm going to talk about, I just want to remind you that one of the reasons, so when we measure alternative splicing, in my case, we're thinking about sort of exon level events, so splicing decisions that the spliceosome makes, rather than quantifying full-length transcripts in our case. And to do this, the only information that really tells you which splice decisions were made are the short reads, when you're using short reads, the short reads that overlap the actual site of intron removal or splice junction reads. And so throughout this talk, I'm going to be referring to this quantity of percent spliced in, which is just the fraction of the reads that tell us about splicing at a particular location that show inclusion of a particular exon. So as you can see, only a small fraction of the reads from a given gene are going to be informative about that. And not all single-cell methods are equal for measuring alternative splicing because the methods that capture just UMI-based methods that capture information near the 3' end of genes are less likely to cover splice junctions earlier in the transcript. And so most of the data that we work with are from SmartSeq-related methods that have deeper coverage along the whole transcript, but at the expense of not being able to sequence as many cells. The methods that I'm going to talk about are equally applicable to other higher throughput technologies, but this is sort of where the richest information can be found. Okay, so the first thing, and this is work that my fantastic grad student, Carlos Bonavent-Najar, wrapped up a few years ago before starting a postdoc last year, was sort of trying to understand what we could learn about the variability of splicing between individual cells. And we, in the course of trying to look at this, we first encountered a sort of big obstacle, and that was that high variance makes it very hard to see differential splicing. So here I'm showing a set of single cells. These are embryonic stem cells differentiated into neurons. And the PCA on the left is arranging the cells by their overall gene expression. You can see the trajectory of their development. And when you overlay the percent spliced in of a particular exon, a particular alternative exon in each of those cells, you see that the splicing from one cell to the next looks highly variable. You have cells that are very similar in overall transcriptome space that seem to have either total inclusion of the exon or total skipping of the exon. And that pattern seems to come up again and again, making people think that splicing in single cells had a sort of bimodal pattern, where even if the overall population average of exon inclusion was let's say 50% in a bulk sample, then when you drill down into individual cells, it seemed like a lot of splicing events showed this binary pattern where each individual cell was somehow only making one isoform or the other. And that was a real surprise because it didn't match what we thought was happening in terms of splicing regulation. And so people noticed this pattern in a number of really interesting papers, and they tried to sort of investigate what could cause that type of consistent sort of state of some sort, perhaps, in splicing outcomes. But we realized in looking deeper into this that this sort of cell-to-cell variability question is a little bit different than it looks at first. And that is because when you start looking deeper into the data, you see that in cells where you actually recover more information, so you have more sequencing reads informing you about a particular splicing event, instead of just seeing this binary pattern where most cells have either a 0 or 1 spliced in inside. If you spread them out by how many splice junction reads you have, you see that the higher the coverage, the more intermediate examples you have. And that's maybe not surprising if you think about the small number of RNAs that existed in the cell to begin with. If there's only 2 of them, there's a limited number of ways you can arrange them. But when we look at genes that have overall higher expression and therefore higher splice junction read coverage for this particular exon, we didn't see that binary pattern at all. And that made us suspicious that the pattern that had been widely observed might actually be related to poor data quality rather than biology. And so we spent a little while looking into this. The model is that if you have a gene with low expression across a population of cells that are overall pretty similar, you're likely to recover, when you lose 90% of the mRNA in making your single cell libraries, you're likely to recover a limited number of isoforms of that gene. Whereas if you have a gene with high expression originally in a whole population of cells, you're likely to recover a higher variability, higher diversity of transcripts. And so this could produce the pattern that we see. And so indeed, when we looked into a single cell SmartSeq data set, we arranged all of the alternative exons by their expression level. Top to bottom is low to high expression, and indeed we see that this bimodal pattern becomes less and less pronounced the higher the expression. So each row of this is sort of like a little histogram on its side showing the distribution of splicing of that particular exon across the population of cells. So starting to look sort of like this might be what's going on. Carlos wrote a nice probabilistic simulator to test these 2 models. I'll skip over the details, but he tried to capture the real patterns of sort of data loss and variability from cell to cell. And he modeled 2 different possibilities. One is that the underlying splicing of most exons really has this unimodal thing with some characteristic underlying percent spliced in that's then lost through data recovery. And the other is that the true underlying splicing is bimodal across population cells. And so when he did these simulations, including a lot of data loss, the real data look a lot more like the unimodal model, where the higher the data coverage, the less of this bimodal pattern you see. But when you have low recovery for exons with low coverage, you do see this sort of artificial binary pattern. One thing that I want to point out, and because where I'm going with this is how we can get around this problem and then go back to trying to understand splicing regulation across single cells. So one important point is that read count alone is not the key here, because especially with SmartSeq type data sets where you don't have unique multimolecular identifiers to avoid PCR duplicates, there are a lot of PCR duplicates. And so what that means is that a high read count does not necessarily mean a high amount of input RNA to begin with or high diversity. And so when we look at the number of reads per cell, it doesn't really tell us much about how good it's going to look. And we had to get around this somehow since we're using SmartSeq with WMIs. We used this really nice trick from Cole Trotnell's group, this census normalization approach that lets you estimate and sort of connect the number of reads that you get to an estimate of the actual number of mRNAs that went into it. And so using that approximation, we could then think about how the number of mRNAs recovered affects this. And so using that census estimate, we saw a pretty good correlation between the number of mRNAs recovered for that gene per cell and the amount of bimodality that we observed in the data. So this gets back to what we think is going on, that when you recover a lot of RNA, you see a lot of diversity. When there isn't a lot of RNA, you don't.
You can continue.
Okay.
The online audience isn't getting audio, so just need to fix it real quick.
So getting back to our problem now, I started by saying that high variance makes it hard to see differential splicing. And we would like to be able to measure splicing changes across single cells without getting confused by this very noisy pattern of cell-to-cell variability. And so when we get back to my slides, I'll show you how we realized if we just do it as a simple first pass filter by the number of mRNAs that we recover, then you can at least limit yourself to high data quality examples where you can recover more accurate information about splicing. And when you do that, as you'll see in a moment, the pattern of bimodality goes away. Excellent timing. And so I hope you all saw that. So when you filter out the cells that have low coverage for a particular
follows a much more predictable pattern across the. So that was a great start, and we used that to show that we can restrict ourselves to only those events and then recover when you then go on to cluster the cells. And of course, you might see some real changes in splicing between different cell types. You might see a true bimodal pattern because there's two very different types of cells, these splicing switches that we've heard about in other talks. But when you cluster the cells into cells of very similar types based on their overall transcription profile, and then limit yourself to high coverage splicing events, then we see that all the exons that remain have a very clear unimodal pattern without this sort of binary phenomenon. So that was pretty convincing, and we convinced other people also. And but it was neat to have sort of pinned this down, but not very satisfying because we still can't find most alternative splicing changes. And we'd rather not have to throw out most data, most genes, if we want to look at splicing. And so Carlos moved on from this to think about how we might be able to model this data problem more explicitly so that we can find alternative splicing that we're quite confident is changing without having to have a sort of arbitrary filter cutoff. The cutoff we used in those slides was about 10 mRNAs per cell for the splicing events across most of the cells. Okay, so this was supposed to be up on Genome Research today, and it might be later if you look, but as of this morning, it's not. So you can see the earlier version on bioRxiv. Okay, so our goal is to find alternative splicing that changes over some landscape of cells in a way that's somehow associated with their identity or state, a differentiation time course between different groups of cells. But we thought it would be nice to do this in a way that doesn't explicitly require a trajectory and does not explicitly cluster the cells, but rather is looking for sort of patterns that change over this space of cells in general. And we want to avoid being misled by the high variance that comes from poor data recovery. And so the approach that Carlos was jointly advised by Nir Yosef, who does fantastic work in the single cell space. And so the idea that Carlos had was based on some other work on transcription from Nir's lab, thinking about the idea of autocorrelation, where you expect the patterns in nearby cells and their overall sort of space to be somewhat similar, and perhaps we can use that to recover something about what's going on. And so if you have just one measurement of a single cell with low mRNA recovery, you have an unreliable picture of the original cell. If you could somehow magically sample that same cell many times, your set of unreliable pictures would combine into one more reliable observation. Obviously, we can't sequence the same cell many times, but luckily we have many cells that are similar. And now that we've sort of shown that the variance between cells is largely due to this technical artifact, we do expect that splicing should be fairly similar from one cell to the next. And so we'll just take advantage of that to look for things that follow that pattern in a way that might show that they're regulated across the space of cells versus things that are just highly variable without a particular pattern. So if you took a population of single cells and you had some sort of splicing that is associated with cell state, let's say these cells are arranged by pseudotime along some trajectory, that regulated alternative splicing might have a shift in exon inclusion from low to high, let's say, across this. And so then when you recover individual cells, even though each individual cell is going to have a noisy, distorted observation, the average from cells near each other is going to show a pattern where, in this case, the cells on the left have a lower probability of recovering the exon included, and the cells on the right have a higher probability. Whereas if you have some alternative splicing, some exon that might have high variance for whatever reason, technical or biological, but is not associated with cell state, then that pattern would go away, that you'd have perhaps equally high variability between cells, but it wouldn't follow any particular pattern where the cells near each other in this space have a sort of consistent trend, that you'd sort of have an equivalent probability of getting one outcome or the other across this whole space. And so Carlos used this to build a probabilistic model of splicing observations and how associated with cell state they are by taking the original distorted observations and then thinking about the average percent spliced in a small neighborhood of cells around each individual cells. So once you've calculated that neighbor average PSI, you can think of the probability that each individual cell, that for one particular exon, the probability of including the exon in this particular cell is going to depend on, we're using the neighbor average as an estimate of the underlying sort of true PSI that we can't observe directly. And then we're also explicitly modeling the number of mRNAs, because there's this sort of binomial sampling process where you're going to lose a lot of information, you're recovering a small number of them. And so there's many different outcomes you could get. So we model that explicitly for our interesting model, model 1, and then we compare that to our boring model, model 0, where there's no underlying pattern. And so for that, we do the same sort of thing, but we estimate the overall PSI based on the global average instead. And then in a sort of likelihood ratio test, we make a score for each exon by comparing those two models for that exon across all cells. So we wanted to see how well this worked. And once again, we simulated splicing data. We used the same model as before, but we added in a sort of change over a time course, simulated some exons with a change, some exons without a change, a lot of complex different types of changes and different changes in abundance at the same time, and then simulate this data loss process so that we have really poor recovery. And when we do that, 6, our method, that's its name, I forgot to say that earlier. So 6 correctly identifies the truly variable exons, the exons that were simulated to have a real splicing change. And so the exon on the left was simulated with a high change in PSI. So you'd imagine that's easy to recover. 6 and other simpler approaches all do very well at that. They also all do well at recovering the exon that was simulated to have a small change in PSI. But but 6, unlike simpler approaches, is very successful at a particular scenario, which is exons that do not really have any change that was simulated to be associated with cell state, but the high variability happens to make it look like there is a change. And so those sorts of false positives turn out to be really prevalent in when looking for alternative splicing changes with other approaches. And so we compared to 2 simpler approaches that have been implemented in some methods and were sort of precursors to this idea. One is the idea of looking at the average PSI between clusters, explicit clusters of cells using the Kruskal-Wallis test. And the other is a plain autocorrelation approach using Geary's C as a statistic that does not explicitly model the sampling process when you have a small number of mRNAs. And so 6 in blue does better across a range of different mRNA capture efficiencies. And in particular, it does better at avoiding false positives. So the figure on the right is the p-values from the null, the data that were simulated to have no change. And 6 does really well at not calling those significant, and these other simpler approaches are very prone to false positives when there was no real underlying change. So then, having simulated all that, we wanted to move on to identify changes in real data. And to do that, it's hard to have ground truth. And so we started with bulk RNA-seq data sets that matched published single-cell RNA-seq data sets. And we sort of lined them up and looked for splicing that changed in this case, this exon clearly changes across a trajectory in bulk RNA-seq, and it also clearly changes in single-cell RNA-seq data. So that would be a positive. And then at the bottom is an example of an exon that does not change in the bulk data, and the single-cell data shows sort of high variance like we've been talking about, but no real change. And so again, we evaluated our method and how well it performed against these, and we also threw in another existing tool for single-cell splicing, and showed that again we were recovering more of the correct things and fewer of the false things, even in cases where the underlying data was particularly full of this problem of like low data recovery and high binary appearing splicing, which is the data set on the right, which obviously has sort of lower performance overall. So that was fantastic. We did all the computational stuff. Now we'd like to move on to see what biology we can see with it. And so here we're turning to a published data set of single cells from mouse midbrain dopamine neuron development. So these were neurons that were sorted and sequenced from embryonic and postnatal mouse stages. And the 3D PCA plot here is showing these cells arranged by their overall transcription profiles. And so we looked for exons whose splicing changed in a way that was associated with this set of cells. And so the scores from 6 of each of the alternative exons that we tested are shown in blue on the right. And we compare that to get a p-value. We shuffle a bunch of data to create sort of random exons to give it a p-value. And so the random exons are shown in gray at the bottom. And reassuringly, these exons were, the exons that we recovered included some that were very obvious splicing changes over this space, and many that were associated with neuron development, had already been experimentally characterized. And so then we thought, okay, well, so here we have a whole set of exons that change across a developmental process, but what we're really interested in is what's regulating those exons. And splicing regulation is complicated. It's mostly done by splicing factors that bind to pre-mRNAs and affect the choice of splice sites. And it can be controlled by one or several splicing factors, and single cell data presents this great opportunity to use the information that you get about splicing factor expression and try to connect that to splicing outcomes. But of course, it's still hampered by this problem of noisy recovery. And so we wanted to move on to infer splicing regulatory networks from the exons that we had identified as significant. So the technical noise might get in the way of discovering co-regulated splicing, even though we've now filtered for sort of exons that have pretty reliable data. There's still a lot of cell-to-cell variability. And so we thought we'd again turn to this neighbor average to look for patterns that were consistent across the space. And we played around with the size of the neighborhood so that we were not capturing, all the cells in one neighborhood, that it should show some variation across the space. And we clustered them based on the similarity of their splicing profiles across all of the cells. And we saw a number of clearly defined modules of different splicing patterns. So I've colored that same PCA, three-dimensional PCA of the cells by the different modules that were automatically pulled out of this clustering method. And you can see that they follow several sort of predictable patterns, things with low inclusion in the early stages, things with high inclusion in the early stages, and also some more interesting ones that sort of at the bottom that sort of go up and then down or have differences in different sort of subregions of this plot. So these are the modules of co-regulated exons. Each module here has at least 30 exons that seem to be behaving in a similar way, and there's a small handful of exons that didn't fit into any cluster. So we wanted to dig into this a little deeper to see if we could connect that to expression of splicing factors that might be regulating it. And we realized in looking at the modules that there was a particular interesting subset of this, which is the mature mouse, the 3-month-old mouse midbrain dopamine neurons. In this PCA, you can see that they sort of separate into 2 clumps. Indeed, those are 2 different sort of biological types of neurons. And we thought we'd look for splicing differences just within that set of cells. And so we took the same thing, we identified the exons that 6 called significant just among this set of cells, things that change, that are different between the different types of mature neurons, clustered them again and found that they form 2 very clearly defined modules with sort of opposite patterns of inclusion or skipping across these 2 sets of cells. And if we took those same exons, so the same exons that defined these clusters in the mature cells, and we look back earlier in development, could we see the point at which these cell types separate and the splicing of some exons separates? Like how far back is that evident? So when we looked back at the postnatal day 1 information, so here each row of this heat map is colored by which of the 2 clusters it was in originally, and you can see that they still cluster together to a large extent, although less clearly than they do in the mature cells. And you still see some evidence of a clear difference in splicing along, really mostly along this 3rd principal component. If you go back to the embryonic stages, the sort of module definitions are less clear, and so there isn't co-regulation of this particular set of exons when you look back to the earliest stages of development, although they might each be being regulated by different splicing factors at those stages. So we wanted to move from just the patterns, like patterns do imply co-regulation, but it's even stronger evidence of co-regulation if you can find something that's co-regulating them. And so splicing factors, when they bind to mRNA, the binding information can be recovered, as many of you know, by the experiments like ClipSeq, where you find the binding sites of individual proteins along mRNAs. So we have information on that from published experiments, including a lot from the ENCODE project, and we also have expression profiles of the splicing factor genes themselves based on RNA-seq data from these same experiments. And so, for instance, the splicing factor NOVA1 that's very important in neurons has a pattern of expression that changes across these cells. So when we look at the correlation between, we first narrowed down our set of splicing factors to things that were enriched for binding to the set of co-regulated exons in these final stage cells. And then we looked for correlations between the expression of the splicing factors and the splicing outcomes. And even though overall, because we're seeing these big developmental changes between these types of cells, there's going to be a lot of things that correlate, but the hope is that we can find the things that sort of correlate at the earliest point that might be causative rather than just correlated. And we're just starting to work on that part of this problem, but as a sort of first pass, we were able to find that these three well-characterized splicing factors all have really strong correlation with the difference in splicing of these two modules in the final mature mouse neurons. That's the things shown on the right. So the correlation between expression of the protein and the splicing of the exons in that module, or each of the exons in the module are the dots that I'm showing there. You can see a clear difference between the blue and the orange cluster. But the interesting thing is that that correlation shows up even in the newborn mice just for the NOVA1 protein and not for the others. And we thought it was particularly interesting that an alternative exon in the NOVA1 gene itself, exon 4, which is known to be autoregulated by the NOVA1 protein, shows a strong correlation with expression of NOVA1 across all of these stages, but its changes in its splicing pattern are most clearly associated in the later stages, suggesting that other factors might be responsible for its splicing at the earliest stages. And so we are excited to keep working in this direction of trying to piece together not just single splicing factor regulation, but also how splicing factors combine in different patterns to produce this full range of outcomes that we see across all of these alternative exons. So this tool is available on GitHub. It's called 6. You can use it. Some people are already using it apparently, which is fantastic, to look for splicing in single cell data. As I mentioned, we've mostly tested it on SmartSeq, but other people have successfully used it on 10x data, and we're continuing to investigate how well it recovers splicing in that sort of sparser information. There should be an acknowledgement slide. Well, this work was done by a number of really fantastic people. Oh, there it is. Okay, a number of fantastic people who are sitting in a tree in this photo, which was our first lab gathering during COVID. So in particular, the work was really led by Carlos Bonabad Nahar, as I mentioned, PhD student who graduated and is now doing a postdoc at the University of Chicago, with some work by Prakriti Bura, a new graduate student in the lab, and this was all a close collaboration with Nir Yosef. And the rest of my lab mostly actually works on translation, but we're really excited to keep going in this alternative splicing direction. So thank you very much.
Great. Thank you, Liana, for an excellent talk. Is anybody have questions to start out with?
Hi, Liana. Hi, here. Thank you for a wonderful talk. And I come from a completely different background, so I apologize in advance if what I'm asking makes no sense whatsoever, right? So you were talking about midbrain dopaminergic neuron maturation, and a question I had for you. So if you go from P1 to P7 to P90, the size of the soma and the size of the nucleus changes several folds. How do you control for the ability, you can recover more exons just because the size is bigger, even if you meet your 10 threshold. So how do you control for that?
That's a fantastic question. And so the quick answer is that we don't control for that. So we are more able to call things significant if there's more mRNA, and that is definitely going to affect what we can call significant. And so one implication is that we shouldn't say anything about the amount of alternative splicing from one type of neuron to the other because we can't really compare that. But we have done some tests just to show a related question that when you have a gene that has a big change in expression along with, which could be either a change in expression or a change in data recovery, that we do reasonably well at recovering things in the face of those big changes in expression, that we're avoiding some of the sort of false positives that can be created just by the you can get a situation where suddenly you have more RNA and it looks like a real change in splicing, but it's actually just that there's the genes being expressed, and we're relatively robust against that sort of mistake.
Can you hear me?
Mhm.
Okay. That was really interesting, and so much to think about. The one thing I was curious about was your sort of pseudotime cell state thing that you had going on, and I thought the regulation was interesting. However, I was wondering if you could talk about, since you could see these patterns from the embryonic stages, what cell types perhaps are being going through that trajectory, and whether it's SVZ or the granular zone. Yeah. Do you know anything about that?
One thing is, these data are from another lab, and I'm not super familiar with neuron differentiation. They were selected to all be dopaminergic neurons, plus other things that sort of came down in that selection. And so I'm trying to remember that there is more information about which markers are separating those two cell types. I just don't actually have it on the top of my head, but we have it more thoroughly described in our paper.
Hi. Yeah, great talk. I was wondering, is there a subset of events in which splicing is truly noisy? Like for instance, cells or events in which there's high expression but are still bimodal, and how well does 6 perform on this subset? Excellent question. And that was, we got into this because we were really interested in cell-to-cell variability of splicing, in particular in the context of the autoregulation that I've worked on in splicing factor expression. So we were particularly interested in those sorts of questions. One thing is that sometimes there are, so we're using transcription profile overall to define cell state, whatever that means. And there are differences that you can see that are more more visible at the mRNA, the splicing and mRNA processing level. So one thing is there can be sort of true underlying differences in cell type that are sort of consistent, but then there's also the possibility of just intrinsic variability, splicing with high intrinsic variability. And that's absolutely the direction I'm most interested in, but we haven't yet really been able to pin down how, can we recover those exact things of splicing with particularly high intrinsic variability. I think that this method would work well at finding those. And so that's sort of a major goal.
I'm going to take one from the online audience and then we're going to switch back.
So we have a question from the online audience. This is from Alexander Falk. Does your averaging model give more weight to cells with better read recovery?
Currently, it does not. So currently, the averaging is just based on everything in a neighborhood being treated differently. But we've played around with different ways of weighting it, and it didn't make a big difference.
Hey, thanks, Liana, for the great talk. I have two questions. The first one is a bit detailed. So you mentioned you chose a few splicing factors that are known to be kind of related to the corresponding exons. What resources did you use to get that information? And shall I?
Yeah, so to answer the first one, we actually started in a sort of unbiased way, and we looked for all splicing factors with enriched binding to within our overall set of significant exons, and then from there narrowed it down to things. So these are things that have binding from published CLIP data sets, and then from there we looked for things with correlated expression. So it pulled out things that are known to be involved in neurogenesis, but we didn't start with that set.
Okay, thanks. And another question, which has been more general. With the technology that sequence long reads or full length mRNA, right? So like the PacBio, etc. Have you thought about using that kind of data to study the splicing patterns, and what kind of advantage or challenges do you see using the long-range data?
Yeah, great question. And we've heard a lot of great talks in this track about that, about how much you can do with long read data. Some of these constraints here are sort of artificial things about short read data, like the fact that you don't recover the full transcript information, but just information about particular events. So far, long read data has much sparser data recovery than short read data. And so the problem of this sort of limited number of isoforms that you might, or limited number of molecules that recover from a single cell, it would be at least as big a concern, probably bigger with long read sequencing, but it could be helpful for sort of pinning down which alternative splicing events we should be looking for to begin with.
How do you identify for a single cell what its nearest neighbors are? Is that based just on expression profile? If if you looked at like using splicing of well-measured genes, does that change what you see?
No, it's a great question. So it's sort of a built-in explicit, not limitation, but framework, right? Is that so what I showed here is all using expression. I mean, we use normalization in selecting highly variable genes, but it's based on expression. And so that means that we might be missing cell states that are really defined by differences in splicing, which could easily happen in neurons because there's a lot of post-transcriptional regulation. So on the other hand, if we used splicing information, then we get a different sort of circularity problem. We have played around with that. We've also considered other ways of defining similarity, even spatial proximity. But for now, what we're showing here is purely based on sort of using transcription as a proxy for cell state, which has some built-in limitations.
So one last question, and we're going to take it from all the way on the other side of the audience here, Liana, over here.
So I was wondering, do you see anything if you look with single nuclear RNA-seq with your analytics scene?
Right. So we've downloaded a number of data sets, and rotation students are processing them, slow going. I think it should work just as well on single nucleus RNA-seq, with the caveat, as people have mentioned, that there are more pre-mRNA reads and it can be harder to count the number of mature spliced RNAs, but it should work just as well on that, with just that caveat that we're going to have a lot of things that look like intron retention, but they shouldn't skew our numbers.